Actually pick up right with myoglobin. So one of the actually nice things about ending the review of the exam with the myoglobin hemoglobin chapter question, because this chapter really focuses a lot on myoglobin versus hemoglobin. Myoglobin is a single polypeptide chain, so therefore it's got a tertiary structure, but there's a kind of quaternary structure in that sense. It does contain a heme. Does contain a single heme. Okay, a heme is a prosthetic group, just like a prosthetic device in a human. It's a group that's present in the final protein, like the active protein, but it's not. It's post-translationally modified. It's added on afterwards, after the ribosome. Okay, and it's not a part of a protein, so it's not like a quaternary structure, like having two subunits come together. It's just an organic compound that's added on that binds iron. Okay. It has no beta sheets. It has 8 different alpha helices, and it's got that stereotypical look where you have the polar side chains on the outside, the hydrophobic core is on the inside. And it's the reason why is because where's hemoglobin? Is it a soluble protein or insoluble? It's soluble because it's there. Does anyone know what does myoglobin do? Well, yes, part of it's in muscles, but what does it do? We're going to talk about the reasons why it's in muscles. It transports oxygen just like hemoglobin. In fact, it does the same thing hemoglobin does. In fact, it's actually more efficient than hemoglobin. But we'll talk about the reason why. And so by the end of this chapter, you should explain why we are hemoglobin-based and not solely myoglobin-based. Okay. And then 2 of the histidines are involved in binding the heme group. And that's due to coordination chemistry, which we talked a little bit about in organic, I guess it was yesterday. But in organic lab yesterday. But what happens is coordination chemistry. We won't go there. But no, so and then there are nitrogens inside the heme group that actually bind to the iron and hold the iron in the correct orientation in order for it to bind 1 molecule of oxygen at a time. And remember, elemental oxygen is O2. But it can also bind other things. We're going to find out it can bind carbon monoxide, carbon dioxide, cyanide, things like that. This is what it looks like. The heme is the heme is relatively planar. There's a little bit of a kink to it, but it's relatively planar, with the iron being held right in the middle of it. And if you notice, there are 2, almost kind of like jacks, where like we played jacks as a child. That's the iron with its coordination. So where it's being held in place by the nitrogens of the heme, and then like the water and an oxygen on it. So I can't tell from here if that one's an oxygen or if it's carbon monoxide that's bound to it. But okay. This is the heme structure. It's a porphyrin, based off of the porphyrin group, which you're going to find out next semester how porphyrin is made. And it's also very, very similar to the perols, which we talk about a little bit in organic chemistry. Okay, and it does matter, the oxidation state of the iron does matter on whether or not how well it binds oxygen. So whether it's iron 2 versus iron 3, for example. And I don't expect you to have to draw the heme, but you can recognize like what the heme looks like. So it's that huge planar compound. All right. So we've already talked about quaternary structure when we have multiple. Oh, that's misspelled. Polypeptides, not peptides or peptides, that go into a multi-subunit protein. And so these are some of the examples that we're going to see throughout organic biochemistry. Let's see, there we go. So for example, we have our good friend alcohol dehydrogenase, which has 2 subunits. And so you should just get practice now, like in learning what the enzyme classes do. So what does the dehydrogenase, what kind of reaction does dehydrogenases do? It does not dehydrate. It doesn't hydrolyze either. It does redox reactions. So, and these will almost, and including this one, almost always uses either NAD, NADH, or NADP, NADPH. And so just redox reactions are done by dehydrogenases or oxidoreductases. It's just that usually the dehydrogenases specifically use NAD, NADH, or NADP, NADPH. Then we have TPI, which is triose phosphate isomerase, right underneath that one. And it's got 2 subunits as well. What do isomerases do? Change the backbone structure from one isomer to the other. And this one, so isomerases change isomers. And what's a triose? We covered that briefly in Organic Chem 2 at the very end of the semester. It has 3 carbons in it, and it is a sugar, and these sugars have to have a phosphate. That's why it's triose phosphate isomerase. Then we have aldolase, which aldolase has 3 subunits. You're going to see that we actually talk a lot about it next semester. It's involved in glycolysis. It's one of the major enzymes in glycolysis. It involves an aldol group. That's why it's called aldolase. Hemoglobin, it says 2 plus 2. It is a tetramer, but it's a tetramer of 2 identical subunits of 2 other identical subunits, but 4 are not identical to each other. So they call them alpha subunit 1, alpha 1, alpha 2, and beta 1, beta 2, because there are no beta sheets. So I don't know why they use the terms alpha and beta, but they do. So that's why a 2 plus 2 means that you actually have 2 subunits and 2 subunits that come together to make 4 subunits. And each group of 2 would be identical. So, all right, lactate dehydrogenase. Once again, that's a redox reaction on lactate. Pyruvate kinase. We've already discussed kinases. It's also a tetramer, but kinases do what? They transfer something. Transfer phosphates using ATP or GTP. This one uses ATP. And by the way, pyruvate is what it works on. And then insulin. Insulin actually has 6 subunits. All right, so the way that oxygen binds, as I mentioned, is a tetramer of 2 alpha chains and 2 beta chains. But once again, they are all alpha helical. There's no beta sheets. It's just they reuse the same nomenclature there, unfortunately. So sometimes we call it the alpha 2, beta 2, or a 2 plus 2. And each chain has 1 heme group. So that means that the hemoglobin can bind up to 4 at any one time, 4 oxygens. Okay, which one is the hardest one to bind? Does anyone know? Of the 4. The very first one. The first step is the hardest, like many things in life. Life integration. Or faith integration is taking that step to go down the aisle to the altar. So once you get down there, it's easy sailing. So similar to that. That's pushing it, but it's as close to faith integration right now as I can think. This early in the morning. All right. Okay, it's called a cooperative binding. One thing that's a little different, and I don't know if you had it in classes, there's actually 2 types of cooperativity. You can actually have positive cooperativity, and you can actually have negative, which I know doesn't seem correct, but they don't call it anti-cooperative. But, so this is positive cooperativity. When that first one's the hardest one to bind, but it causes a conformational change in the neighboring structure, the neighboring subunit, which makes it easier for the second one, which makes it easier for the third, which makes it easier for the fourth. Okay, and then once all 4 are bound, it's completely, you can't bind any more than that. Okay, and the function of hemoglobin is to transport oxygen, and the structure of the oxygenated, and by the way, I forgot to point it out, but HB is just an abbreviation for hemoglobin, MB is the way they abbreviate for myoglobin. The structure of oxygenated hemoglobin is actually different than unoxygenated, and since they're all alpha helical, what happens is when that first oxygen binds, it causes a shift in a couple of the alpha helices, which sort of like a little ratchet, because then that causes a shift in the neighboring one, which makes it easier for the oxygen to bind, which causes a shift in the neighboring one, so it's like a little domino effect. Okay, there are also other cofactors that can help make it either easier or more difficult for the oxygen to bind hemoglobin. H+, which is the fancy way of saying pH, right? So pH is just a concentration of H+. Carbon dioxide, not carbon monoxide. Carbon monoxide will affect the oxygen binding, but it's because it won't allow oxygen to bind at all. Chloride anion, which I don't know if the other lines came through. And then BPG, which you're actually going to find out, we discuss BPG a lot next semester because of the whole interplay with glycolysis. And at this point in time, I don't require that you know the structure of BPG, but it's something that you actually could, well, if it's going to work, could actually figure out, because if you just look at the root word, which is glycerate, which comes from glycerol, and so what does glycerol look like? Right, so, well, not fatty acid chains, it's 3-carbon backbone with alcohols, it's a polyalcohol. So I deliberately didn't put the other hydrogens in yet, because that would be glycerol. So glyceric acid, which is just a carboxylic acid form of it, would look like this. Well, I don't know if the little oxygen came through, but it's a carbonyl, it's a carboxylic acid, which if you remember from organic Chem 2, that by default makes that carbon number 1. Glycerate just means that it's lost the carboxylic acid's hydrogen, so it becomes a glycerate. And then 2,3-bisphospho or bisphosphate, I mean that's what it is just add the O. It means there are phosphates there instead of the hydrogens. Which I'm going to just do the cartoony looking because I ran out of room already, but this is not phosphorus, that's a phosphate. And so then you can put the other hydrogens in. So that's what it would look like. That's BPG, 2,3-BPG. Okay. And so you're going to see the glycerol and all of its derivatives over and over and over and over again between this semester and next semester. And so that's why you should know what glycerol is, and then you can always work backwards to figure out glycerol to glycerate, which means you made it. Right, and I don't want to give anything away. But we also have, if you have a bisphospho, then you can have a monophospho, which they don't call it monophospho, but it'd be like 2-phosphoglycerate or 3-phosphoglycerate and things like that. But we're going to see it over and over again. This is so important. And so we get to go talk about math here, but using math logic. You don't need to memorize the numbers so much, but you need to understand what the shape of the curves mean. That is like, I can guarantee you this is going to show up on quizzes and exams about these shapes. So you're going to see this kind of thing over and over again. Maybe even come across it when you study your enzyme. This is a binding curve. And so there are different ways that they show this. Sometimes they do it as a percent instead of degree or that's the degree of saturation. I would also call it a fraction of saturation because you can go from 0 to 1. If it's percent saturated or percent bound, it goes from 0 to 100 if you're doing percent. Okay? And then it's very similar to what we're going to call Km later on, or Kd. So what happens here is we can see that, which is actually more efficient at binding oxygen, myoglobin or hemoglobin? Myoglobin. In fact, it can approach the asymptote of 100% bound, or the fraction of 1. Whereas hemoglobin actually, under all circumstances, actually doesn't become 100% bound. And it's sigmoidal, which is just a fancy way of saying what kind of curve is a sigmoidal curve? I mean, obviously, it comes from sigma, but what does that indicate? What letter of the alphabet does that look like? S. It's an S-shaped curve. Okay, and so I would expect you to be able to draw that. What is this shape called? Or, I mean, what kind of curve is that one? Banana? No. Boomerang? No. That is a hyperbolic curve. And so, whereas the hemoglobin is sigmoidal. It's an S-shaped curve. And so at any concentration, so what they do is you always have fraction bound or percent bound on the y-axis. You have concentrations, whether this is in torrs or molar, it doesn't really matter. Like, I don't want you to memorize the numbers, but I want you to know the shapes of the curve. And we talk about a way that you can shift the curve. So when we add certain cofactors, it may make it less or more sigmoidal, or it can shift it left or right, things like that, which becomes really important. So make sure you can read this graph. Then they like to use, sometimes it's called the KD, not KT, as in a katydid or as the name, but KD, as in the dissociation constant, For the fraction bound, that's the concentration when 1/2 bound or 50% of it's bound. So that's why they've got this little line here. And it shows you the relative KD, or the parent KD, is right around 25-ish torr for oxygen. And myoglobin, it's really, really tight. I mean, so tight that I can't hardly see it. I mean, it's less than 5, like 2.5 or something like that. Once again, the number's not that important, as in the shape of the curve. So, my question for you is, why is hemoglobin our primary source of oxygen and not myoglobin, since myoglobin combines it so well?
Myoglobin, the connection is a lot stronger, so it's harder to release oxygen.
Right. Myoglobin is that stingy kid in the playground that won't share the toys and holds on to it until the last possible moment. And so then what happens is you would die because you would have to get to such low concentrations of oxygen in your tissues before myoglobin would give up its one oxygen. Whereas with hemoglobin, we can all just take a deep breath in. And so we get all the oxygen into our lungs, and we exhale out, and we exhale out the carbon dioxide. And so what happens is by changing that concentration, the concentration of oxygen in your lungs gets really high, way up here, then a high fraction of the hemoglobin is then going to be will bind to the hemoglobin. The hemoglobin then travels to the muscles, the brain, the other tissues, and where the concentration of oxygen is low, let's say it's around 10, and then you're only going to have, and I don't know if that's the exact number, because it's going to depend upon what you're doing and what tissue it's going to, but then instead of being 90% bound, it's down to 5%. Only 5% of the hemoglobins would still have the oxygen. Whereas at that percentage, if we were myoglobin, yes, it would bind all of the oxygen. In fact, it would be even better at binding the oxygen from your lungs, carries it to the tissues, but then you would have to literally become hypoxic before it would release that one oxygen. Okay, so that's the reason why we aren't. Now there are, yeah.
Why do your muscles use myoglobin?
So okay, get to that. Why would your muscles use myoglobin? And when do they use myoglobin? Whenever you're exercising, because what happens when you're exercising? You're pumping that iron. You're running. I always say you're running from a bear. You're running from that bear. I don't know why. Which, by the way, little side note: if you're running from a bear, you always run downhill. You don't run uphill. Does anyone know why? This is one of the little facts I read from biology. Well, it's because of their body shape, and so since they're so heavy, it's like they fall more easily. Whereas running uphill, they've got the stronger body shape, so they can outrun you going uphill. But running downhill, they're more likely to fall. So, and don't climb a tree, bear. Yeah, it's not like I'm in cartoons. But yeah, so you're running from that bear, running really, really fast. So what happens to your muscles? You get oxygen depletion. And so then you actually do approach that hypoxic thing, and it takes too long then for the hemoglobin to go to the lungs, come back, go to the lungs, come back. And so therefore, it's like a reserve. It's like your reserve thing for that burst of energy to give, the myoglobin will give it all up, and then also that the hemoglobin has time to go and exchange the carbon dioxide for oxygen in your lungs to replenish it. And then later on, you can replenish the myoglobin. In fact, myoglobin can, in theory, take it from hemoglobin because it has a higher affinity, right? So the odds are that once it gets released, any empty myoglobin right there could take it back. So, okay, and we're going to actually talk about some of the diseases that affect hemoglobin's binding, or some different tissues that have different proclivities towards binding oxygen. Okay. All right. Now, and I don't remember this part either, but I know that there are certain fish, I'm pretty sure it's fish, that have a much higher concentration of myoglobin than we do. And the reason why would it happen with deep water fish? Well, not that big. We're talking about really big differences in pressures for the oxygen and stuff, and so therefore they can let it go and bind it and stuff much more efficiently. But I don't remember which fish, and I don't remember the relative percentages, like if it's just twice more than what we have, or it's a higher percentage or not. Okay, so this comes to the whole idea of allosteric enzymes, which in organic chemistry we talk about steric hindrance and how steric strain, how this bumping and grinding between molecules. Well, allosteric can be both positive or negative, but don't get that confused with steric hindrance. Okay, this is where cooperativity comes into effect. Okay, so the allosteric behavior is the existence of multiple forms for that quaternary structure of the enzyme. And so there are a couple of different models that are like the two extremes. One's sequential and one is, oh my lord, I can't think of the name of it right now. It's something my sequential versus, I'll come to you in just a moment. Okay, so you can have an allosteric effector, and so that's something that modifies the quaternary structure. It can either be positive or negative, okay? Usually when we're talking about allosteric, we're talking about positive cooperativity instead of a negative one, but they can be. Homotropic modulators can be activators, and so that's whenever the binding of the normal substrate, which substrate is just the enzyme word for a reactant. It's what the enzyme acts on. Whenever the binding of its normal substrate or ligand, a ligand just means anything bound. Okay. And what happens? So this is like oxygen. This is, for example, oxygen binding to hemoglobin. That first one makes it upregulates or activates it to make it more likely for the second one or the activity to occur. All right, so usually we're talking about things that are upregulating. Another example that, I don't know in the most recent edition of your book that they use, but is ATCase, which is aspartate transcarbamoylase. Or aspartate to ATCase or oxygen to hemoglobin. Heterotropic can be either activators or inhibitors, but that's when it's some other non-substrate or a non-normal ligand that actually can act as can kind of fine-tune it, either upregulate it or downregulate it. Okay? So when we'll see with some of these examples, is it reminds me of, here in this classroom, you can imagine that it's either, activity like this light switch, but it's either on or it's off, on or off. Whereas, I've got one of my light switches at home is a little dial, so you can kind of have the mood lighting. Except what's funny is the previous homeowner hooked it up to the ceiling fan, so you can either turn the ceiling fan down really low or turn the ceiling fan up really high. But it's sort of like that. You can regulate that, and that's how some of these work. Is because depending upon what ligands are bound to it, it can either crank it up closer to that 100% power or decrease it down, and so you can have fine tuning by what's bound at that time. So once again, ATCase here, CTP inhibits it, but ATP activates it, and oxygen, it goes on, there's lots and lots of things. And then we've already talked about oxygen binding hemoglobin, but what happens is we'll find out that 2,3-bisphosphoglycerate actually affects the way that oxygen binds to hemoglobin, and it shifts that curve one way or the other. And so by doing that, you can, if it's present in the tissue, then you can either make it more or less bound to the hemoglobin. Okay, concerted. Why can't I think of that term? So concerted versus sequential. So there are two models that talk about this allosteric enzyme. And what does it, well, first of all, if we just start with sequential, what does that mean? One after another, First this happens, and second this happens, blah, blah, blah. Where if it's concerted, if something's a concerted event, what happens? Pardon? So, no, if it's a concerted action, it happens at the same time. It's like the whole concert piece coming together, so to speak, if you want to think of that, like the parts of the orchestra all coming. And so, now, these models rely on the fact that you can have your enzyme and your protein exist in two states. One state's called the T state, and one's called the R state. And I've heard R's usually stands for relaxed. Like relaxed, it's cool, so it can bind the ligand or the substrate more easily because it's relaxed. T is either tight, tense, or taut, T-A-U-T. And so I've heard all three used. So whichever you can think of it as tense versus relaxed, it's fine with me. It's just the T state. The T state is less likely to bind its substrate or ligand, right? Because it's all tight, it's tense. Okay, whereas the R state, it's relaxed, it's chilling. All right. So, for the concerted model, what happens is the R, the relaxed state, binds really tightly, and that's binds the substrate or the ligand. Because like hemoglobin is not an enzyme. It's a worker, but it doesn't do a chemical reaction. So it's a transporter. So usually, a lot of times I mention enzyme, in the book mentions enzyme, but you can do the same idea with hemoglobin, and technically it's not an enzyme. All right, but what happens is in the R state, something can bind more tightly than it could in the T state, so that's considered like the active form, whereas the T, tight, tense, or taut, it binds less tightly, it's inactive. In the absence of the substrate, most of the enzyme would be predominantly, there's an equilibrium going on, so most of the enzyme is going to be predominantly in the inactive form, the T state. However, what happens is whenever adds the substrate causes a change in the equilibrium to come on over and form the R state. So what we're saying is, let's say that we have something that, in the simplest, it has to have at least 2 subunits. So what happens is, we've got two T's, they're both tense, but whenever you start to add in the substrate plus substrate, then it changes this over to R's. Okay. And so, therefore, it's in the presence of substrate, it shifts. Well, actually, there we go. And so, I'm not going to have enough room to draw it all out on this one, so I'll just do it like the next one. Okay, so changing from T to R and vice versa, all subunits change simultaneously because it's concerted. It's a concerted action. It's going at the same time. So that's why I was trying to show up the T's and the R's, but once I put in inhibitors and activators, you'll find out that I didn't leave myself enough room. Okay. So here, and I don't know why this book was the old book, and it didn't use KD. It used L. Instead, but it's supposed to be K, like equilibrium constant, or KEQ. I don't know why they used L for ligand, I guess, but that's, we don't usually use L. We usually say, whoops. Oh, I know that. Thank you. Oh, come on now. We would say the equilibrium constant is the concentration of T over R. Okay. And so what happens is in the absence of substrate, it's really high in favor of the T state. However, once you add that substrate, since it binds on the binding side, it's going to, whoops, let me do that. It's going to force the equilibrium down. And so by forcing equilibrium down due to the Chatelier's principle, it's going to pull some of the parts that had been in T over to the R to replace it. So that's why you add substrate. It's going to take some of it from the pool that was already existing in the R state, and just since you're removing some of that product, you're going to cause it to convert over from the T state to the R state. Okay. And you can also have things that are called activators. So if we imagine, if we had an activator that preferentially bound to the the R state, then we can even have it add another end, I should say. And so, I'm not going to be very good. This is R with an activator. And that makes it even easier for it to bind substrate. Plus that. So now we have R, A, S. So that's what an activator does. Okay? It pulls the equilibrium over again and make it even more likely to bind substrate. And it'll take even more from the T side to make it up due to the Chatelier's principle. Okay, so at this point, I'm going to end right here with the concerted method.